afternoon. Living systems inevitably form ecosystems. Therefore, in order to understand the origin of life, we need to understand the origin of ecosystems and their stability and how ecosystems interact with their planetary environments. So this is a paragraph from Astrobiology Roadmap proposed by NASA. What it says is that astrobiologists have long recognized that in order to understand how life can colonize a planet and how life can have enough abundance to show its observable biosignatures, we need to understand the formation and the maintenance of the whole communities, especially microbial communities. For example, bacteria and their predator viruses and their interaction with the environment. So as we have learned from Everett Schock yesterday, we know life is a planetary response. Planets get trapped in metastable states, and the life helps them get out of that. So in other words, the purpose of life is to help a planet to approach equilibrium. And in principle, about the process are also driven by the thermodynamic constraints. But life can outcompete biochemistry by finding new and faster metabolic pathways to utilize free energy gradient. And how does life do this? Life does this by controlling information flow. So one of the things that distinguish life and non-life is that life can control information flow and use information flow to manipulate and impact its environment. So when we talk about information flow, it can be gene flows, and usually people think gene flow from mutations, but later we'll see that there is a much more powerful mechanism for gene flow, which is horizontal gene transfer. And horizontal transfer is a mechanism where the genetic functionalities can be swapped between different organisms. So there are other different ingredients for life. For example, a living system is an ecosystem, and it has two aspects. One is population dynamics, and for example, the behavior of predator and prey in an ecosystem through birth and death processes. And another aspect is metabolism, where metabolic network inside organisms will form a global electron market that transduces the energy flow. So these two aspects has to be unified, but so far we don't have a framework for that. So in this talk, we will focus mostly on the aspects of population dynamics with a little bit of energy flow, while the next talk will focus on metabolism, but no population dynamics. And also, we will look at the population dynamics of marine microbes and their interaction with viruses. And the energy flow will come from the photo ingredients. So this is a simple diagrammatic summary, which says that a living system can outcompete chemistry by utilizing gene flow, metabolism, and population dynamics. And this is the outline of my talk. So the system we're interested in is the marine cyanobacteria and their predator viruses called cyanophage. And I will show that horizontal gene transfer between cyanobacteria and the cyanophage will form a collective network that drives the whole ecosystem to evolve. And I will show that by build up a minimal model, and the results will explain the range expansion and niche stratification that have been observed in real data. Surprisingly, we'll see that predation, in this case, actually will stabilize the ecosystem and is beneficial to bacteria. So let me quickly explain the spirit of our modeling. So conventionally, when people talk about bacteria and phage, they are thinking of prey and predator. So they model the system by using differential equations, for example, like Volterra type of models. However, like we explained before, in a unified way, including energy flow and information flow, the interactions and relationships between bacteria and the viruses are much more complicated. So later I will show detail how we incorporate these ingredients in a model. So the scientific organism we're looking at is called Prococcus. It's one of the most abundant organisms on the world, and it has very small genome size. It's highly streamlined. For comparison, E. coli has about 4 times larger of its genome size, and it fixes a large amount of carbon on Earth. Especially, unlike most bacteria, Prococcus has no immune mechanism like CRISPR or prophages. So how does it avoid extinction under viral attack? It turns out Prococcus find its defense against viruses by changing its surface protein, where viruses would attach on cells. And it appears to be a special signature in its genome called genomic island, and we know it's probably from horizontal gene transfer, and therefore it shows a huge pangenome. So a pangenome is the whole collection of genes of the same microbial species. And the Prococcus has a very large pangenome, and is much larger than any single organism in the world. And that means that any 2 Prococcus cells would share only very few genes in common. So on a much larger scope, Prococcus has another feature, which is niche stratifications with 2 emerged subgroups. For example, here on the left panel, it shows light intensity as a function of depth in ocean. And there are 2 subgroups, the 2 ecotypes of Prococcus. One is high light adapted, colonized near the surface of ocean. Another one is low light adapted ecotype. And our model will try to explain this phenomenon. Also, Prococcus phage, called cyanophages, carry photosynthesis genes. And it has found that cyanophages with photosynthesis genes actually have higher burst size when they do the lysis. And it turns out the cyanophage got these photosynthesis genes from horizontal gene transfer. So this is a phenology data found by Penny Shizen's group. And this is published in the paper by Sullivan et al. In 2006. So here it shows that in the middle of a bacterial gene for photosynthesis 2 genes, the virus genes appear in the middle of the bacterial genes. So this is the evidence of horizontal gene transfer, that viruses got genes from bacteria. So their interpretation for this data is that bacteria and viruses can form a collective state and create a global reservoir for photosynthesis genes that benefit both bacteria and viruses. And in this talk, we're going to show by a minimal model how this can actually work. Okay. So in our model, the mechanism for horizontal gene transfer is done through the process called generalized transduction. So in generalized transduction, a virus finds a bacterial cell and infects it and inserts its genome into the bacterial cell. And it will hijack bacteria's machinery and so make bacteria to produce many copies of viruses, and the bacteria will die. But occasionally, very few of baby viruses would accidentally grab some bacteria genes. So these genes won't function like normal viruses. Instead, they will more like zombies. So when one of zombie viruses float and find a new bacteria cell and infect the new bacteria cell, this new bacteria cell won't die, and instead they can utilize the bacteria genes from the previous bacteria. So this is how the bacteria can benefit from a viral predation. So how does that horizontal gene transfer can work collectively? This is how the story can work. So here, this color area shows a biosphere of bacteria and viruses. And the plus sign here, for simplicity, stands for the beneficial genes that, for example, provides more efficient metabolism. And the minus sign stands for genes that are less efficient. So first, viruses can get bacteria genes from horizontal gene transfer. And the key point here is that viruses has higher mutation rate. So therefore, the viruses are like a generator of new genes. And they create either better genes or worse genes, and they dump these genes back to bacteria. And now bacteria got the new genes from viruses. And another key point is that bacteria here do streamlining. So they behave like a filter. So they filter out the bad genes, and only bacteria with beneficial genes can survive. So by this way, the bacteria will save the beneficial genes and will transfer these bad genes back to viruses. And the virus would get the better genes. And this process would repeat again and again. And in the end, the whole biosphere communities can get better and better. So next I'm going to show a sequence of models by adding a step-by-step different evolutionary mechanisms to explain how the previous cartoon picture can actually work. So first, I'll consider a very simple model where bacteria and viruses have a type of infection predation gene, and I will show how horizontal gene transfer will have its effect. And later, I'll consider a different type of model where bacteria and viruses have another type of genes, which stands for photosynthesis efficiency. And we'll see the effect of horizontal gene transfer as well. And for the next model, I'll consider the case where the genes have deleterious mutation. And in the end, I'll include the photo gradient as the energy flow. Okay, so this is the first model. The first model is very simple. We consider bacteria and the viruses as prey and predator. And here, the ecological processes here are like the chemical reactions. So here, bacteria, we assume bacteria and viruses have a type of genes whose value stands for the ability to defense or attack. So here, for example, the big B with subindex I stands for a bacteria individual with some gene value I. And the birth process here is that this bacteria has some probability B per unit time to produce another same copy. And another process of competition means that a different bacteria would compete with the same food resource, so one of bacteria would die. And both bacteria and the viruses would die naturally with some probability. And if in predation, one bacteria encounters viruses, if the virus carries a gene whose value is larger than the gene value of that bacteria cell, there's a probability P that this bacteria would be killed, and an amount of phage burst size of baby viruses will be produced. And the problem of this model is that usually the phage burst size M here is large. It's about 10 to hundreds. So we would imagine that the viruses could easily overwhelm the whole population and drive the extinction of bacteria. And this is what we observe in our simulations. So this is a population of bacteria and viruses. The red is viruses, the bacteria is in blue. And we see that the population of viruses would oscillate a lot and finally will drive the population to extinction. However, life finds its own solution. In reality, we know that genes can mutate. So we consider mutation of the genes. For example, when a bacteria with gene value I give a birth to another copy, there is an additional probability mu b here for the new copy to mutate into a different gene values. Okay, so and same for the predation. So for predation, there's an additional probability mu v that the baby viruses would mutate into different gene values from its mother. So in this case, we found that both bacteria and virus genes can co-evolve. So this shows gene value, the average gene values of bacteria and viruses as a function of time. And we can see both of their gene values can evolve and increase. And it turns out in this case that both bacteria and the viruses can co-evolve and avoid the extinction. So, however, there's another problem. The problem is that usually the bacteria has slower mutation rate than viruses. So in this case, the viruses would mutate much quicker than bacteria, so its gene values grows faster with time. In the end, that means that the virus will become stronger and stronger and eventually still outcompetes bacteria. Again, life finds its own solution. So if we consider hydrogen gene transfer, like a general transduction process we explained before, so these two processes will describe the gene flow from bacteria to viruses and viruses to bacteria. And viruses with high mutation rate can keep generating new genes, and they can transfer these genes to bacteria and eventually form a flow of beneficial genes. And that beneficial gene flow will help the bacteria which have a slower mutation rate. So in our simulation, we see again the coevolution of the genes in bacteria and the viruses, and it turns out that both bacteria and the viruses can keep their populations and the ecosystem becomes stable again. So here we consider a second type of model. Previously, we considered bacteria and viruses have a type of gene stands for infection, predation. But now here we consider bacteria and the viruses have a different type of genes, and that gene stands for photosynthesis efficiency, photosynthesis ability. So for example, this bacteria B with subindex I stands for bacteria with some gene value, photosynthesis gene value I. So it can, for simplicity, we set that the birth rate of bacteria is a function of its photosynthesis value. So different bacteria would have different birth rate and to create a new copy. And similar for viruses. So viruses have a photosynthesis gene J would have a different birth size that depends on that gene value. So with higher gene values, that viruses would have a higher burst size. So in our simulation, similarly, in the case that the virus has higher mutation rate, the viruses would quickly evolve into very high burst size, and the population will eventually overwhelm the bacteria population. So it will drive the bacteria to extinction. So again, if we consider horizontal gene transfer, then now the viruses can produce good genes and transfer this beneficial gene to bacteria. Then bacteria can also increase their burst size by utilize viruses' genes. So in the end, the photosynthesis genes in bacteria and the virus can co-evolve together again, and their population can be sustained. So next, we are going to consider a different case. So in general, for well-adapted species, most mutations are neutral or deleterious. So deleterious genes from mutation accumulates, and that will make individuals die, and the population will shrink. In that case, it's usually called Muller's record. In this case, when population decrease, the demographic stochastic would be more important, and leading population go to extinction faster. And we think that in range expansion, that individuals expand its colony. At the front, the population is small, so demographic fluctuations are important. So this effect would be significant. So we consider this effect in our model of photosynthesis genes. So here we plot the photosynthesis gene of bacteria and the viruses function of time. Because of deleterious mutation, the virus with higher mutation rate would decrease their genes very quickly, and in the end, their population would collapse. But if we turn on the horizontal transfer, then the bacteria now can help viruses. We can see that by looking at the distribution of photosynthesis genes in bacteria and distribution of photosynthesis genes in viruses. So these black lines stands for the genes transfer from viruses to bacteria, and here the black line stands for the genes transferred from bacteria to viruses. And we see that the genes transferred from viruses to bacteria can range over many values. However, the bacteria behaves like a filter. They will keep only the genes which are beneficial and transfer back to viruses. So viruses now can have more frequencies at the beneficial genes. So it turns out, because of horizontal gene transfer, now the photosynthetic genes in bacteria and the viruses won't decrease, they won't degenerate, and then they can co-evolve, and their population can be stable in this ecosystem. So we can study the phase diagram of this model. So here we plot the ratio of viruses with beneficial genes as a function of horizontal gene transfer in the units of mutation rates. So in principle, we can change the horizontal gene transfer rate by changing the density of the population of the individuals. Here in our simulation, we can do the horizontal transfer rate, and we found that only when the horizontal transfer rate is large enough, then the bacteria and the virus can form a collective state. So there will be enough beneficial, enough population of beneficial genes in viruses. However, we also found that the horizontal transfer rate cannot be too large. If it's too large, that means that all phages would already have very good genes, and more horizontal transfer will just add the less beneficial genes into a population. So that will give the system a negative effect. So here I want to address again why this is a collective state. So we found that in our simulation, it is very important that we have both ways of hydrogen transfer from bacteria to viruses and from viruses to bacteria. So therefore, they can keep the beneficial genes in both bacteria and the viruses. So in our simulation, if we turn off either one side of hydrogen transfer, for example, at this time point, the system will quickly just collapse. So this shows that this is a truly collective state that requires both ways of hydrogen transfer between bacteria and viruses. So finally, we include the energy flow by adding a background of photo gradient. So this is the data I showed you at the beginning. And in our simulation, we also create a background of photo intensity as a function of depth. And here we include the photo intensity into the birth rate of bacteria. And also, the death rate of bacteria depends on the photo intensity because the cells near the surface would get burned. Okay, so in your model, we first start with low light adapted ecotype that evolved first. So they initially occupied at the bottom of the sea. And then they coexist with the viruses, and they can do hydrogen transfer with the viruses. And we turn on the simulations, so eventually we can see the development of this low-light adapted bacteria and evolve into a different ecotype, which is high-light adapted bacteria. But the key point is that if we don't have hydrogen transfer, it takes very, very long time for the low-light adapted bacteria to mutate into the high-light adapted bacteria through just a pure random mutation. So only through hydrogen transfer, we can see a relatively reasonable timescale that this evolution of ecosystem development could occur. So I will show you an animation of our simulation where the yellow stands for low-light adapted bacteria at the beginning, coexisting with the red viruses. And eventually they will form high-light adapted bacteria, which is in blue color. So here we initially have low-light bacteria coexist with viruses, and they do hydrogen transfer, and the viruses diffuse with bacteria and help the low-light adapted bacteria develop into high-light adapted bacteria. And we can see the distinct distribution of two different ecotypes. So our simulation results explain that cyanobacteria and phage can help each other. Bacteria can get help from viruses because viruses have higher mutation rates, so they behave like a generator of new genes. So bacteria can get beneficial genes from viruses. And viruses can get help from bacteria because bacteria do streamlining and has slow mutation rate. So bacteria can help viruses to filter out bad genes and keep only beneficial genes. So in these ways, they can help each other and form collective states. So here I like to summarize what this our minimal model can explain. So the results of our model are consistent with the facts in Porcococcus. For example, Porcococcus has highly streamlined genome, and that limits the metabolic redundancy. So the only way to improve metabolism is to improve the efficiency. And we know, and that means that we know there's no immune mechanism like CRISPR prophages in Protococcus, and that's because bacteria don't need that. The cyanobacteria can balance between the risk of predation and the benefits from horizontal gene transfer. And that will reflect in the huge pangenome in Protococcus and the presence of genomic islands. And it also data shows niche certifications, which is a result from the viral mediated utilization of the energy flow. And also, our model shows that cyanophages, which carry photosynthesis genes, can help improve photosynthesis genes in the bacteria, and that's consistent with the fact in Protococcus. So here are some take-home messages. So at the beginning, I showed that the planetary living system theory will require a unified picture of in ecology, which there are two aspects. One is population biology and energy flow, and we still need a unified framework for that. And in this talk, I use mostly population dynamics and a little bit energy flow. And also, ecosystems are collective. And our model and simulation shows that sometimes the ecosystem stability should be understood by considering multi-scale interactions that ranges from the scale of genes to the scale of the whole environment. Another key point is that information flow, especially through horizontal gene transfer, that provides a gene flow that allows life to maintain and expand its range in thermodynamic gradients. So with that, I will stop here, and I'll be happy to take any questions. Thank you.
All right, thanks for the great talk, and let's open it up for questions.
So the discovery of photosystem 2 genes in phage around 15 years ago was very curious. So as I mentioned earlier in a passing comment about D1, so D1 protein is the PSBA protein, which turns over very rapidly. It also has one of the fastest promoters of any gene known in a eukaryotic cell. So do you think that the viruses co-opted these genes in order to promote their own rapid reproductive capability within their host cell? Or is there some other reason that you can imagine that these genes very specifically were incorporated into phages?
So the questions is about the details of evolution of photosynthesis system genes.
But their incorporation of those genes into viruses, into marine viruses, was never, when it was discovered, it was thought it was an artifact. It's obviously not an artifact. I just wondered if you have any thoughts as to why those genes would be in a virus. They're very abundant.
Right. So let me try to understand the question. Are you asking that why those genes have to appear in viruses?
Okay, so maybe I'm not clear. So in photosystem 2, there are a series of genes in cyanobacteria and every alga and every plant that splits water that encodes for this one protein called the D1 protein, the diffuse band 1 protein, and the gene is photosystem PSB, photosystem 2, subunit A, right? So it's PSBA. All right, now, PSB, yeah, okay. So now when we get to this gene, It turns out this protein product of the gene turns over very rapidly, about every 25 to 30 minutes in the day when the light is shining. So in order to keep that repair mechanism going, the cell makes a lot of extra D1. So it has a promoter that is very, very, very highly demanding of RNA. So it's a very rapidly turning over system. So we use that promoter a lot in molecular biology to promote other genes. I mean, we just cut the business end of the gene off and use the promoter. So I'm just wondering if if you don't have a positive feedback in the system here, that the virus essentially selected or uses that gene from a selection point of view in order to promote its own burst size, its own facilitation of its own growth within a host cell once it's invaded. You're following my logic here? Yes. So I'm just wondering if there's some feedback here that you could think of. I mean, is that you? I'm trying to understand why horizontal gene transfer of PS2 genes is occurring so much. Those genes. It's not Rubisco genes. It's not, it's not just any random gene here.
So I think the feedback picture you're talking about, we can kind of see that in this country picture, right? So let me quickly repeat again. So once the bacteria got some beneficial genes or more efficient photosynthesis genes from viruses, the bacteria do streamlining. So that means they keep only small size of genes. Small size of genomes. So bacteria with less efficient genes would just die out. So therefore, only bacteria cells with more efficient genes can stand out during selection. So that will give, when they do whole gene transfer and dump this gene back to viruses, this will give a feedback effect that increase the genes in the biosphere of viruses. I think that's like the feedback effect that you mentioned.
It's curious.
So
Many other cyanobacteria have multiple copies of the photosynthesis gene. They're different. Other cyanobacteria have multiple copies of the photosystem 2 gene. So I think it's Synechocystis has 3 copies. Two are different from each other, and they're expressed differentially depending on the stresses. Rogier, I mean, is there more than one copy in Prochlorococcus?
When they do this whole gene transfer, if through the generalized transduction, they will dump a lot of genes, not just photosynthesis gene, but also the genes that can code the surface protein that will help them defend the viruses, and that will have its benefits through whole gene transfer, and that will reflect in the diversity in bacteria. It's not just one gene.
yeah, beautiful work. I have a question that perhaps bridges a little bit what Paul is talking about and extensions of these ideas that I was wondering about. Which is, have you thought about, for example, having other types of organisms around, other phytoplankton or other bacteria, where possibly in sort of rare circumstances you could have cross infections between the phage, transferring genes across species being a font of innovation? And the reason I ask that is that in the evolution of Prochlorococcus, it's actually not just photosynthesis genes that are being transferred, but all the major divergences within the lineage at the sort of macroevolutionary timescale look like they're horizontal gene streams coming from different groups. So in connection to what Paul is saying, it's actually not just photosynthesis genes for which this sort of thing seems to happen. Yeah. And I'm just wondering about the role of phages in those kinds of transfers.
Yes, I believe there are different examples that show similar mechanisms.
Right.
So, for example, there are different types of cyanobacteria that not live in ocean but live in rocks. So they would form different layers of rocks, and they will utilize the different intensity and frequency of light depends on the depth of the rock layers.
Yeah.
Yeah, and we also believe there might be similar mechanism in SARS-11.
Right.
Yeah.
So there's a particular gene you might find interesting to look at, which sits in the electron transfer chain,
Yeah.
called the plastoquinone terminal oxidase, and that looks like it's being transferred between viruses of different
Right.
phytoplankton groups.
Yes, for bacteria with very small genome size, and we would guess we could see similar effect that, for example, there might be evidence that we can find some genes in bacteria that come from viruses, and that would be an example that this mechanism can work. Yeah.
So just to go back and answer your question and Paul's, and unfortunately Paul is just leaving, but what's special here about the photosynthesis genes is that both the bacteria and the phage can utilize them in their life cycle, whereas other genes that are horizontally transferred from bacteria to bacteria, the phage is just the bus that takes them from one place to the other. But what's special, and what is the feedback that Paul was asking about, is the fact that both the bacteria and the phage are able to directly utilize the photosynthesis, the PSB genes in their life cycle. So that's what gives you the nonlinear feedback that stabilizes the system.
So we should talk more because there are other genes for which that may actually hold.
Okay, yeah, exactly. Then as Hongyan said, there's other systems where we know there's other genes that can do that too. They can yeah.
Other questions? We have a couple more minutes.
So it's not a fair question because it's not well thought out. But as I listen to the talk, I have a question that's much like when we listen to chemistry talks. When we listen to the chemistry talks at the beginning of the meeting, we have a problem of getting from some starting point to some molecule. And most things we try don't work, and eventually we find a solution that gets there. In population processes, if you take the properties that you think are native properties of bacteria and you think are native properties of viruses, and you plug them into a model, the model goes unstable and everyone dies. And you change some details and it goes unstable and everyone dies. And eventually you work and you find a model that can produce stable population processes. At the end, you tell a story where there's a kind of a general set of principles that you think are at work, that viruses handle some jobs, bacteria handle other jobs, and it's a collective state that produces stable populations. How much can you show that even if a particular model is wrong, those principles are likely to be the real reason you have stable populations in the world? And maybe some other model that's different than the one you thought of, but that's organized by the same principles is still the correct explanation.
Okay, let me try to understand your question. I guess you are asking that if this mechanism is robust enough, or is just requires many different conditions to make it work. Okay, so we think this mechanism is robust. First, we can predict the phase diagram, and that phase diagram is consistent with the data, and we can show the phenomena in real data. And it's not, it doesn't require fine-tuning of parameter in our simulations.
Right.
So which means the result is stable. And we might be able to think of different kind of models that maybe different type of bacteria with different mutation rate. But that way, in order to see the same effect, the same phenomena in real, like what I show here, the niche stratification and the collective effects, we also need some other conditions, like which would be too manipulating, like if it's not bacteria and viruses, like in these real ecosystems.
Right.
So we think it could be. I think it's a robust mechanism for this ecosystem.
It has automatically homeostasis for the ecosystem built in, whereas other proposed mechanisms for the stability of phage systems, like low dimensionality or things like this, those really don't have homeostasis emerging naturally. You have to do some fine-tuning of parameters. As Hongyan said, here, without it generically stabilize the state, it's not the only mechanism that can do this. But in this particular system, it seems to be the one that is appropriate because of the utilization by both biosphere and bacteriosphere of the photosynthesis genes.
All right, that puts us right at time. So let's thank the speaker again.